Я могу уже говорить? Дима, а чего это вот первый слайд не сменяется вторым? Так, презентация, где тут презентация, подробнее, добро пожаловать, сообщение. Я что-то не вижу тут опции презентация. Ага. Мне вот это принять, да, нажать?
Да. Один раз нажми.
Да? Извините. Мне вот на верхнюю зеленую кнопку или слева зеленую? Верхнюю или нижнюю? У меня тут две принять. Слушайте, опять что-то, наверное, разрешить следующей программе внести изменения, что-то я не то нажал. Да, разрешать? Хорошо. Так. То есть я могу, да? Ага, хорошо, хорошо. Спасибо большое. Спасибо, Дим. Все нормально. Да, да, спасибо большое. Угу. Спасибо. Good afternoon, ladies and gentlemen. Topic of today's lecture is bacterial genetics.
Поздоровайся.
Good evening, good afternoon. Thank you for coming. Well, in this first slide, you see on the left upper part structure double-stranded DNA. Into the central drawing, you see circular double-stranded DNA of the chromosome of the prokaryote bacterium, and in the right upper part, you see the third drawing. This is a chromosome of the eukaryotic cell. Bacterial genetics. This is an active research area studying heredity and variability, resemblance and difference between parents and their progeny. In the slide, you see here two cells, daughter cells, immediately after formation of cross wall, after replication of these cells. The flagellated cells of the Pseudomonas aeruginosa. Like other origins, bacteria also mean their characteristics constant from generation to generation. It means both the daughter cells must be absolutely identical. But at the same time, bacteria exhibit variations. Variations is a particular property in a small proportion of their progeny. Variations, it means we will say today about genetic material that maintains stability of the genome, and about variations. When saying about variations, we will say about genotypic variations or phenotypic variations, among genotypic about mutations and recombinations. The central dogma of molecular biology is applicable for bacterial genetics also. The essential material of heredity is desoxyribonucleic acid, DNA. Genetic information can be, this genetic information may be duplicated in the process of replication, transcribed onto the messenger RNA, translated to the proteins. It means that DNA, when replicated, serves the template for synthesis of the daughter progeny DNA molecule. DNA molecule serves as a template for synthesis of messenger RNA. Messenger RNA, after binding to the ribosome, serves the template for synthesis of the protein. Arrangement of bacterial genetic material. Arrangement. The genome of the typical bacteria is a total collection of genes carried by the bacterium chromosome. But in this drawing, you see chromosome molecule of the bacterial DNA in the form of large purple circle. Exception for the chromosome is this cell you see, only one chromosome. Bacteria may contain so-called extrachromosomal genetic elements. Also, extrachromosomal genetic elements are named mobile genetic elements. Extrachromosomal genetic element in this figure is shown by the red ring. This is a so-called plasmid. Additional circular molecule of DNA additional to the chromosome, and you see green bacteriophage that injects to this bacterium dark green molecule of the DNA. This is also extrachromosomal genetic material. It means material that may occur separately from the bacterial chromosome. The list of the extrachromosomal genetic material consists of plasmids. Here you see circular red plasmid. Transposons are not shown. Integrons, they are not shown. Transposons are smaller than plasmids, and integrons are smaller than transposons. IS sequences. IS is insertion sequences. They are sequences of nucleotides that are smaller than transposon and plasmids. And bacteriophages. Of course, not the entire bacteriophages, but genome of the bacteriophage. Bacteriophage DNA. It means genome of the bacteria consists of the chromosome and extrachromosomal genetic mobile elements, namely plasmids, transposons, integrons, IS sequences, and bacteriophages. A few words about distinctive characteristic of these elements. Chromosome. Chromosome encodes all information essential for the normal bacterial life and functioning of the bacterium for its growth and multiplication. So, chromosome contains DNAs that code for all the essential processes that involve in the growth and multiplication and the normal life of the bacteria. Extrachromosomal genetic elements or mobile genetic elements are not essential for bacteria. It means bacteria may survive without these extrachromosomal genetic elements. They are additional elements. But these extrachromosomal genetic elements may encode properties leading to survival advantages of bacteria under the proper conditions. For example, such the advantages may be resistance for antibiotics that may be established by synthesis by the bacteria that have the resistance plasmids, for example, proteins capable of destroying some antibiotics. For example, beta-lactamases that may be encoded by the plasmid or transposon genetic elements help bacteria to survive the presence of antibacterial drugs. Migrating elements such as plasmids promote bacterial evolution through horizontal gene transfer. Horizontal, it means from one cell to other, not from the maternal to the progeny. This vertical transmission, but from one bacterium to other that occurs in the same time to nearby bacteria. They provide an important source of genetic diversity and play a fundamental role in bacterial ecology and evolution. It means extrachromosomal genetic elements are instruments of the bacterial evolution, directed for better surviving of microbes in the environment that may change their properties. Arrangement of bacterial genetic material. The chromosome can exhibit quite dynamic reorganization following the insertion or deletion of transposons, integrons, and genomic islands under fluctuating evolution environment and selective conditions. No? Chromosomes can exhibit quite dynamic. It means chromosome may be changed due to the integration in the structure of this chromosome of the some mobile genetic elements, ectochromosomal genetic elements, such as the transposons, integrons, and genetic islands, what new term, genetic islands. We'll discuss what is this later. Transposons briefly, are linear pieces of DNA, often include genes for antibiotic resistance. Transposons can migrate between unrelated plasmids and or the bacterial chromosome independently of the normal bacterial recombination process. It means transposons are genetic elements capable of transposition. It means jumping genes from chromosome bacterium to the plasmid, from one plasmid to another. The next element, integrons. Integrons form an essential building block of many transposons and allow the rapid formation and expression of new combination of genes in response to selective pressure, no pressure of the environment. Bacterial chromosomes. Of course, they are most important, essential DNAs of the bacteria. Bacteria need to have chromosomes to survive. Usually, bacterial chromosomes or bacterial chromosome is a single supercoiled circular molecule of double-stranded DNA that has molecular weight about 2000 million daltons. Daltons, this is a unit of weight of the one atom of the hydrogen. Single circular double-stranded DNA chromosome contain many bacteria, for example, enterobacteria. No, really, there are Shigella species, Salmonella species, Escherichia coli, Klebsiella, Yersinia, and other enterobacteria. Well, they have one circular DNA molecule of the chromosome. Chromosome composed of about 5 million base pairs, or 5 kilobase. 1 kilobase is 1,000 base pairs. Bacterial chromosome is haploid, has no introns and no histones. Contains about from 2 to 3,000 genes. Genes are the segments of DNA carrying codons. Codons are codon sequences that may code for particular polypeptides. Bacterial chromosome has a single replicative origin. Single replicative origin, this is a part of the DNA that is required for binding to the bacterial chromosome of the DNA polymerase that is required for synthesis of the copy of the chromosome before replicating, in the process of replicating of bacteria, before dividing of this bacteria. Of course, we understand that is replicative origin in the bacterial chromosome would be mutated, broken or changed. Bacteria after that would be incapable of replicating. Exceptions. All the criteria you see here have the exception. Exception either, for example, Vibrio cholera, causative agent of one of the most known diarrheagenic bacteria, causing very severe diarrhea. Vibrio cholera has not one but two circular chromosomes, totaling 4 million base pairs of DNA sequences and 3.9, almost 3.9 thousand genes. More than what 3,000 shown up. In this slide, you see structure of the genome of the Vibrio cholera. No, two cells of the Vibrio cholera shown in the right part of the slide, and every of the cell has large chromosome and small chromosome. Small chromosome and large chromosome both have place of the part of the molecule that is named replication origin. It means if one replication origin in large chromosome would be broken, replication region is the small chromosome will help bacteria to produce at least copy of the small chromosome. These two chromosomes are circular, and both contain parts of the DNA structure responsible for so-called virulence factors. Virulence factors are the instruments of causing of disease in the pathogenic bacteria. What in the large chromosome occur what CTXF factor. CTXF factor, this is a sequence of nucleotides responsible for synthesis of the subunits of the cholera toxin. A cholera exotoxin, this is a powerful virulence factor, the main virus factor of the cholera that is possible for causing of diarrhea. In the small chromosome, chromosome number 2, you see red part of this DNA that is named integrin island or pathogenicity island. This island contains genes responsible for additional virus factors, for example, for some proteolytic enzymes and some adhesive molecules that have the Vibrio cholera. So Vibrio cholera is not only example of exclusion. With Vibrio cholera, Leptospira interrogans, Brucella abortus, and other species of Brucella have two circular chromosomes that consist of the single supercoiled double-stranded DNA. It means some bacteria have one circular DNA chromosome, some other have two. But some bacteria, for example, Borrelia burgdorferi, have the not the circular but linear chromosomes. What here in the upper left part of the slide you see linear structure of the bacterial chromosome of the Borrelia burgdorferi. No, you see in the red photograph that Borrelia burgdorferi is a spirochete, motile spirochete that may be transmitted through ticks from animals to humans, causing so-called Lyme disease. Exception for linear chromosome, Borrelia burgdorferi has plasmids, and these plasmids may have linear and circular structure. It means some plasmids also may occur in the linear structures. Well, brief characteristics of bacterial chromosome concerning function of different elements of the chromosome. Genes. What the structure is named gene. Genes are sequences of nucleotides that have a biological function, such as cisterns. Cisterns are responsible for synthesis of certain protein. In the right part of this slide, you see theoretical structure of bacterial chromosome, or maybe plasmid. You see that some of the parts of the circle have the origin blue and purple color. And near to these structures, you see mRNAs. Orange mRNA, blue mRNA. What genes are with fragments of DNA that may be transcribed to the certain messaging RNA that codes for the certain protein or certain polypeptide. But here you see structure of aperon. Aperon, this is a complex of genetic elements that are responsible for coordination of the functioning of the genetic structures that are parts of this aperon. What do you see here? You see upper part of this aperon, this is a red molecule that is named promoter, or promoters. Promoters are regulatory genes. Promoter, this is a regulatory gene that can code for the protein but serves as the place in the molecule of DNA that may bind to the RNA polymerase. But if promoter is open, is activated, enzyme RNA polymerase binds to the promoter, and if promoter is open, RNA polymerase moves along the molecule of the DNA. When this RNA polymerase binds to the structural gene number 1 with orange, result would be synthesis of mRNA. RNA polymerase synthesizes messenger RNA, the template of this DNA orange gene. After that, RNA polymerase comes to the second structural gene of the blue color, and the mRNA is synthesized. New mRNA is shown blue, and this mRNA after would be translated to the second protein. Purple structural protein in this apeiron also serves as the DNA coding for protein and also would be transcribed and translated later. When RNA polymerase finally come to the what's last element of this operon, so-called transcriptional terminator part of the operon, RNA polymerase is detached from this DNA, and the process of synthesis of proteins that encoded by the operon may be repeated or may be blocked if promoter would be blocked. But how promoter in this operon may be blocked? Block, it means incapable of binding RNA polymerase and capable of beginning of the process of the transcription genes that are parts of this promoter. So promoter may be blocked if absolutely other gene that is named aperon, also regulatory gene, produces repressor proteins, for example, that may bind to the promoter and prevent binding of RNA polymerase to this aperon, shown here. In this case, operator gene will regulate transcription of genes that are shown here, the part of the promoter. It means structural genes code for proteins, promoters and operators. Promoters and operators are the regulatory genes that control so-called expression of genes. That's parts of the aperon. Expression, this is a protein synthesis of proteins that is encoded in a certain part of the genome. Aperon, you see, so aperon, this is a group of one or more structural genes for this DNA molecule that code for certain polypeptides. Genes expressed from a particular promoter and ending at the transcriptional terminator. How the function of aperons are regulated? In this slide, you see process of the expression of the so-called lac aperon, lactose aperon. Maybe you know how does work this aperon. I only have to repeat briefly. So, in the upper part of the slide, you see linear structure of the regulatory gene LAC1 and blue, green, and yellow parts of the promoter of this lactose aperon. White structure that looks like the white leaf stump, for example. This is RNA polymerase. Normally, regulatory gene, like one gene, when is transcribed, so the template for synthesis of the messenger RNA. This RNA is translated, and finally, like one gene is expressed. It means protein synthesized. This protein is named repressor of the lac aperon. This red repressor, when is unchanged chemically, structurally, usually binds to the part of the promoter that may specifically bind this repressor. This process is shown in the upper part of the slide. If red repressor binds to the yellow part of the promoter, RNA polymerase with this white fish cannot move along the apeiron elements and can achieve the lag Z. Lag Z, this is a structural gene that codes for beta-galactosidase. A beta-galactosidase, this is an enzyme that may chemically destroy lactose in the process of the nutrition and respiration. So, upper part of the slide demonstrates how expression of the lactose apeiron may be stopped. What is happening? But pay attention to the lower part of the slide. In the lower part of the slide, you see a situation when bacterium occurs in the environment contain lactose molecules shown by the green balls. Lactose, when is uptaken by bacteria and transferred to the cytoplasm, react in the cytoplasm to the repressor protein. What this red repressor protein binds molecule of lactose, and lactose modified spatially repressor protein, and this repressor protein modified can react to the promoter. And if promoter becomes open, RNA polymerase moves along this promoter. After that comes to the lag Z structural gene, lag Y, lag A, and other structural genes of lactose apeiron. These genes are transcribed and with very long pink polyprotein is synthesized. After that, this polyprotein is cut into the distinct functional proteins. For example, lag Z that calls for beta-galactosidase. When is translated, activates synthesis of beta-galactosidase. So this is a sugar-lytic enzyme that ferments, involved in fermentation of lactose. It means chemical degradation of this molecule. This molecule, when it's digested by beta-galactosidase, disappears, and new repressor proteins, when synthesized, have the structure that helps them to bind to the promoter and stop process of the transcription translation from the structural proteins. By the way, beta-galactosidase, this is an enzyme that help us and bacteria to consume lactose, digest lactose. LAC-Y gene codes for permease, a permease that serves the pouring protein so that lactose may easily migrate from the environment to the cytoplasm of the bacteria. But here you see how is regulated synthesis of lactose apeiron. In this slide, you see how does the process of the expression of the tryptophan apeiron. Tryptophan apeiron is responsible for the destroying of the nutrient, such as the glucose, lactose, and other sugars. A tryptophan apeiron is responsible for synthesis of this amino acid in the bacterial cell, for example, in the Escherichia coli. In the left lower part of our slide, you see structure of this amino acid tryptophan that is important in the process of synthesis of the many proteins. So, what do you see here? Again, tryptophan apeiron. Tryptophan apeiron consists of promoter and structural genes, tryptophan E, tryptophan D, tryptophan C, B, and A. In the upper part, you see a situation where tryptophan R repressor gene is transcribed to the mRNA. This translated to the protein, and this protein also is protein repressor. But if tryptophan in this cell, free tryptophan in the cytoplasm is absent, repressor occur in the inactive form and can bind to the promoter, and RNA polymerase in these conditions causes transcription. After that, some translation is happened, of the structural genes. These genes code for different enzymes involved in the synthesis of amino acid tryptophan in this cell. When concentration of tryptophan in the cell becomes high due to the activity of the tryptophan apeiron, molecules of tryptophan bind to the protein, repressor protein, change the conformation of this protein, and this protein, as a complex to the tryptophan, becomes capable of blocking the tryptophan apeiron, preventing activity of the RNA polymerase that attach to this fragment of DNA molecule. After this blockade, of course, structural genes responsible for tryptophan synthesizing enzymes becomes blocked, and synthesis of tryptophan in the cell is finished. This is an example of the negative regulation. Negative when product of the that is encoded by certain gene or group of genes becomes blocked in the presence of this protein, of this amino acid, this amino acid. Brief characteristic of bacterial genome. No, this slide is devoted to the history. The first bacterial genome was sequenced in 1995. It's natural to associate the advent of the modern era in the genomics with the appearance of the first complete genome, namely the genome of the pathogenic bacteria named Haemophilus influenza. It means first complete genome sequenced in the genetic laboratories discovered the structure of the Haemophilus influenza. This is a gram-negative bacterium causing gastric duodenal ulcer in humans. The second genome that was sequenced among the pathogenic bacteria was the genome of the Mycoplasma genitalium. This is a bacterium causing chronic infectious disease of the genital organs. Now many bacterial genomes are discovered, not only this. In this slide you see theoretical structure of the circular genome of the Escherichia coli. You see that different elements responsible for different functions of the genome are marked by the numbers from 0 to 100. And we can use this labeling in the process of mapping of the genome to identify where is localized apeiron responsible for the certain function. What you see that lactose apeiron is situated near to the 8th unit, characterizing the special distribution of the Escherichia coli genes. What tryptophan apeiron, what this tryptophan apeiron, is present near to the between 27 and 28 units that measure the size and responsible for mapping of the genes in the Escherichia. In this slide, you see also structure of the circular DNA, chromosomal DNA of the Staphylococcus aureus. Here you see that what blue core gene structures of this circle, consisting of essential genes, are supplemented in certain parts by the additional genes. Now, first of all, replication origin, what is situated in the upper? This is a 0 element of the gene. But near to this replication origin is situated resistance island. This is a so-called pathogenicity island, consists of genes that code for resistance for some antibiotics, and they are mobile genetic elements for these genes coding for resistance against a certain proteins. Pathogenicity islands is not only pathogenicity islands. Other pathogenicity islands is situated the right direction of the this genome. By the green color are shown places of integration to this staphylococcal genome of the different bacteriophages. You know that bacteriophage is the virus of the bacterium that has DNA genome, and this DNA genome of many bacteriophages may be integrated to the larger DNA molecules of the bacteria. And this map demonstrates at least 1, 2, 3, 4, 5, 6, 7 sites of the situating of the prophages, bacteriophage genomes integrated in the structure, the core structure of the DNA of this bacterium. Mobile genetic elements. Mobile genetic elements are extrachromosomal DNAs of the bacteria. They were found by Barbara McClintock. This scientist was awarded by the Nobel Prize in 1983 in physiology and medicine for her discovery of mobile genetic elements. Later was found that mobile genetic elements play a critical role in the spread of virulence factors such as exotoxins, exoenzymes, and some other genes of the bacteria that are responsible for pathogenicity. Pathogenicity, this is the capacity of causing disease. Strategies to combat certain bacterial infections by targeting these specific virulence factors and mobile genetic elements have been proposed. It means now scientists try to change some mobile genetic elements so to cause the integration in the bacteria with the damaging of the structure of this bacteria to limit spread and replication of pathogenic bacteria. In this slide, you see mobile genetic elements of the Escherichia coli. What is the central part of this drawing? You see commensal Escherichia coli. You see yellow cytoplasm of this bacterium, blue DNA in the circular chromosome, and this is a commensal bacterium. Commensal means bacterium incapable of causing disease, incapable of the destroying of the structures of the host, of the human. But near to this central bacteria, you see 1, 2, 3, 4, 5, at least 5 pathogenic variants of the Escherichia coli. They are strains, they contain additional genetic elements. But for example, the left pathogenic bacterium is named enterotoxigenic Escherichia coli. This Escherichia has an addict in the form of the green plasmid contain straight transposon for enterotoxin. Enterotoxins are the exotoxins, protein exotoxins that may damage epithelium of the intestinal tract. It means that enterotoxigenic Escherichia coli, due to presence of the enterotoxigenicity plasmid, tox plasmid, it's called for enterotoxin, acquires the pathogenicity and capacity to cause the toxic damaging of the human epithelial cell. Lower, you see enteropathogenic Escherichia coli. This enteropathogenic Escherichia coli has additional plasmid, PEAF, so-called. This plasmid is responsible for synthesis by this bacterium of the adhesins that strongly attach this bacteria to the surface of epithelium of the small intestine, causing destroying of this epithelium that finally causes malnutrition in the children affected by this enteropathogenic Escherichia. Below the basic commensal molecule, commensal structure of Escherichia coli, in the lowest part of the slide, you see intrahemorrhagic Escherichia coli. In this intrahemorrhagic Escherichia coli, you see structure of bacteriophage, not the entire phage really, but genome of this bacteriophage. This phage, bacteriophage genome, contains phage STXF toxin that contains genes for Shiga toxin. It means this bacteriophage CTXF genetic elements of the virus cause for very powerful Shiga toxin that may cause destroying of the human epithelial cells and enter hemorrhagic Escherichia coli. Hemorrhagic means that this bacterium, due to the presence of this Shiga toxin and capacity to secrete this toxin, may cause the profound destroying of the epithelium of the large intestine, causing hemorrhagic colitis. No, enter-invasive Escherichia, what you see, invasin plasmid. Also have the plasmid of invasins. Invasins are the special proteins that help the enter-invasive Escherichia of the spreading from one damaged human cell to other human cell, maintain invasion, process of the spreading of the bacteria in the layers of the human epithelial cells. And the last, uropathogenic Escherichia coli, and you see here red arrows, pathogenic islands. But these bacteria have so-called pathogenicity islands. What do you see here? You see edicts, the certain groups of genes that are accumulated in certain parts of the bacterial chromosome. They are integrated molecules acquired from different origins outside of this bacteria that accumulate genes in these islands that code for different toxins, attachment proteins, and other virulence factors of bacteria. Uropathogenic Escherichia are hemolytic. It means in some of these pathogenicity islands occur genes that code for hemolysins. The toxins capable of causing destroying of the red blood cells. Now what do you see here? You see how on the basis of the non-pathogenic Escherichia could be formed 1, 2, 3, 4, 5, at least 5 groups of the pathogenic Escherichia that are named differently, cause different pathogenic effects and different diseases in humans. One word about pathogenicity islands. Pathogenicity islands, they're shown here, here, and here. It is 3 zones here. And what do you see? Pathogenicity islands are part of the flexible gene pool or sets of genes that encode traits that may be favorable under certain environmental conditions. Pathogenicity islands possess the following characteristics. Briefly, pathogenicity islands are segments of the bacterial genome, not essential segments of bacterial genome, that accumulate genes that code for pathogenicity factors that may maintain disease. Such factors are the toxins, different toxins, exotoxins, adhesins, molecules that attach bacteria to the human cells, invasiveness factors that help bacteria to spread in the human body. But these islands may grow because newly acquired external genes of the pathogenicity may be attached to this, may be integrated inside of these pathogenicity islands. And these islands are a demonstration of the pathogenicity of bacteria that carry these islands. What are the distinctive properties of these pathogenicity islands that are sets of the extra genes for bacteria? Properties of these islands are presence of virulence genes that code for toxins, adhesins, invasins, and other. Second, specific presence in pathogens. Pathogenicity islands, if are present in bacteria, demonstrate that this bacterium is pathogenic. these alleles are absent in benign relatives. It means these genetic elements are absent in the other strains of the same bacteria that may be commensal. Pathogenic alleles are large, distinct chromosomal regions from 10 to 200 kilobase. Characteristic of these pathogenic alleles have the base components different from core. What I have to explain. Usually, certain bacteria may be differentiated from other species or member of other genus of bacterium by many methods. One of them is calculating of percent of the guanine and cytosine in the genome or in the genetic element that you discover. So all the essential elements of genome of the bacteria, they have the percentage of guanine plus cytosine typical for this bacteria. But pathogenic islets are the foreign elements that integrate to the core of the bacterial chromosome may have and usually have other percentage of guanine plus cytosine content. Atypical for core, it means basic supporting structure of the genome, typical for this bacteria, for the virulent variant of this bacteria. The next property of pathogenic isolate, frequent association with chromosomally integrated mobile genetic elements. Pathogenic isolates usually are associated to integrated plasmids, integrated to the bacterial chromosome transposons, IS sequences. Why? Because plasmids, transposon, and IS sequences are mobile genetic elements that bring with these pathogenic genes and integrate them to the core structure of the bacterial chromosome or bacterial plasmid. These mobile genetic elements serve as the delivery system to bring the new pathogenicity gene for toxin and integrate this gene to the structure of the chromosome of the bacteria. Pathogenicity is characterized by genetic instability. It means that may easily frequently change their structure in different environmental conditions. And they have mosaic structure of several acquisitions. It means pathogenicity islands are sets of the genes that code for traits that may be favorable under certain conditions. For example, favorable if bacterium infect the human body and factors that help this bacteria to survive in the human body of the patient. Plasmids. Migrating genetic elements, plasmids, the second plasmid. In this drawing, plasmid is shown as a red circular molecule of DNA, and by the arrow, this molecule is shown as a addict that may be integrated to the purple bacterial chromosome. Plasmids are extrachromosomal double-stranded circular DNA molecules, which can occur in cytoplasm in the free form, free from the chromosome, or gram-positive, gram-negative bacteria. Plasmid can replicate autonomously, independently of the bacterial chromosome. It means plasmid may produce progeny molecules quicker than chromosome may produce copies of the chromosome. Plasmids can integrate, or this process is shown here, integrate into the bacterial chromosome. And in this case, plasmid will replicate together with bacterial chromosome because plasmid in this case will depend on the regulatory genes of the chromosomes that regulate chromosome replication. Plasmids range in size from 100 to only 5 genes. It means plasmids may vary in the size. Due to these, plasmids are classified into transmissible large plasmids or non-transmissible small on the basis of ability to transfer among bacteria. It means transmissible plasmids may make the cell of forming special F pilus. Through that, copy of the plasmid may migrate to the neighboring cell of the same species or related species of the same genus. Transmissible plasmids. But here, transmissible plasmid is shown with a small yellow plasmid in the green cell, yellow circular DNA against the black background, and a red circle drawn in the right photograph, demonstrating presence of the ring plasmid, synthesis of the linear copy of this plasmid that may migrate through the pilus, the whole pilus, from the upper cell to the lower, and copy of this plasmid migrate from upper cell to the lower. This process of the spread of the plasmids in the population of cells is named by the word conjugation. Conjugation is a mating, very analog of the very primitive sexual act. So transmissible or conjugative plasmids, or conjugative, that may be transmitted by this conjugative process, can be transferred from cell to cell, or from upper cell to the lower, by conjugation. Conjugation is a process of mating. Transmissible plasmids are large, molecular weight from 40 to 100 kilobase. Transmissible plasmids contain so-called F factor or fertility factor. F, it means fertility. F factor codes for genes for own transfer of the F plasmid from one cell to other. Transmissible plasmids carry genes encoding information of new properties of bacteria, such as antibiotic resistance. It means if F factor in the plasmid is combined to the resistant gene that codes for penicillinase, but beta-lactamase enzymes destroy an antibiotic, in this case, conjugation may give to the recipient concerning this DNA cell, gives additional properties. This cell, after transfer of the F plasmid to this cell, becomes F positive. It means acquires this plasmid, and this cell can mate to the neighboring cell, other neighboring cells, transform to the F positive cell. And if antibiotic resistant genes is attached to the F genetic factors, the donor cell acquires additionally with this resistance for this antibiotic. Transmissible plasmids are usually present in a very few, from 1 to 3 copies. No, we understand that if certain cell may produce copies of the plasmid, these copies activate cell to transfer these copies to the neighboring other cells in the process of the conjugation. And bacteria pushes out these new copies of genes, transferring them to the other cells of the bacterial population. Non-transmissible plasmids. Non-transmissible plasmids can be transferred from cell to cell bulk conjugation because these plasmids don't have F genetic factor. Due to this, usually, non-transmissible plasmids are small, molecular weights from 3 to 20 kilobases, not 400 as for conjugative plasmids. Non-transmissible plasmids are frequently present in many from 10 to 60 copies per cell. Now understand, if they replicate, accumulate in the cell, but can be transferred, the concentration of this plasmid becomes higher and higher. The third position. Non-transmissible plasmids don't contain the transfer genes. It means they don't have F factor. F factor, this genetic element of the bacteria that calls for what capacity to mate, capacity to form peeling proteins from pilus through the whole of that channel, plasmid may migrate from one cell to other, from positive of positive cell to the negative. And non-transmissible plasmids can be mobilized for transmission by conjugative plasmid, bacteriophage, or host chromosome. What does it mean with this last position number 4? It means that if non-transmissible plasmids occur in the bacterium, they may fuse, integrate to the conjugative plasmid, and as the part of this conjugative plasmid, this initially non-conjugative plasmids combined with the conjugative may be transferred to the other cell in the complex, to the conjugative. Bacteriophages also may uptake non-conjugative plasmid and involve in their genome, and if bacteriophages cause lysis of the cell, some of them may be supplemented with the non-conjugative plasmids uptaken from this killed host cell. Let's see. Plasmids, exception for size, exception for capacity to be transferred in the conjugation process, may be classified on the basis of the functions that are encoded by these plasmids. But here you see 7 variants of properties that may be encoded by plasmid. Antibiotic resistance, for example, is typical for so-called R plasmid. R means resistance. Bacteria that have these plasmids have the genes coding for proteins that in different ways prevent lysis of bacteria in the presence of the antibiotic. Against that, this plasmid is active. Second, exotoxins. Some bacteria have plasmids that code for exotoxins. Now, we talked about this before. What do you remember slide with the Escherichia? What you see that bacteriophage CTXF contains genes for Shiga toxin, very powerful exotoxin. What bacteriophage gene in the structure of hemorrhagic bacteria Escherichia coli make this bacterium pathogenic. And this plasmid that may contain genes of this bacteriophage would be plasmid of toxicity. In this slide, other example of the exotoxin encoded plasmid is shown. Chlai. Chlai plasmid, this is a plasmid of the staphylococci that is responsible for synthesis of the hemolytic exotoxin. Hemolytic exotoxin causes hemolysis, destroying of red blood cells. What in this slide you see a fragment of petri dish with a blood agar. Right, the central elements of this agar are inoculated with the bacteria. You see many small colonies, and these colonies are surrounded by the yellow transparent zones. These zones are yellow because this is the color of milk-peptone agar. Why are they not red? Because red blood cells are destroyed. Chlai means hemolysis. Hemolysis plasmids are responsible for hemolysin synthesis. Hemolysin are exotoxins, majority of them. Third type, bacteriocin producing plasmids. For example, coal plasmids. Bacteriocins are the bacterial enzymes that destroy other bacteria, usually of bacterial species related to the species producing bacteriocin. It means Escherichia coli may code for colicins. What plasmids that code for colicins is named coal plasmid. Escherichia coli. What this col, it means colon, intestinal tract. And it means that even Escherichia are hosts of these plasmids. What coal plasmids of Escherichia codes for colicins. Colicins are Escherichial enzymes that destroy Salmonella, for example, or destroy Yersinia. In the contrast, Pesticins are bacteriocins of the Yersinia pestis, causative agent of the plague. Pesticins are directed to eliminate and destroying of the other enterobacteria, including Escherichia coli. It means bacteriocins are instruments of the competition of bacteria that pretend to colonize the same habitat in the human body or environmental habitat. Fourth, resistance. Resistance to heavy metals, mercury and silver. Fifth part of the plasmids. Plasmids responsible for resistance to ultraviolet light. Now, resistance to ultraviolet light is encoded by cities of pigments that prevent penetration by this light of the cell wall of the bacteria. It protects the genome of this bacteria from ultraviolet light. Pili or fimbria. Pili also may be encoded by the pili encoding plasmids. And the last, antibiotics. Some bacteria have plasmids that code for antibiotics. For example, antibiotics produced by Streptomyces. Streptomyces are the bacteria, some strains of that are produced antibiotic streptomycin, very powerful antibiotic, destroying many bacteria, including mycobacteria, including Mycobacterium tuberculosis. You see here, finally, so 7 variants of the products that may be encoded by the bacterial plasmids. On this slide, you see a structure, theoretical structure of the resistance plasmid capable of maintaining conjugation, conjugative R plasmid. This is a circular structure that's typical for many plasmids. A red portion of this DNA structure is responsible for resistance transfer factor, RTF. We know that transfer factor, this is a F factor, resistance transfer F, fertility factor. It means this is a, from one side, F plasmid that may cause own transfer to the other cell in the process of the conjugation. Blue elements of this plasmid, this, and this, responsible for resisting against certain antibiotics. For example, erythromycin, penicillin, chloramphenicol. Green elements of these plasmids are also responsible for resistance, but resistance not for antibiotic, but to toxic substances, arsenic, antimony, and some other substances. What resistive mercury, resistive for lead. Small molecular weight substances may be very highly toxic, some heavy metals including. It means plasmid that you see here has at least 2 different functions. One, with F plasmid, this plasmid is conjugative and may make cell having this plasmid of transferring this plasmid to other cell in the process of conjugation. It means cell having this plasmid is F positive. From other side, this is a resistance plasmid that help bacteria to survive in the presence of some of the antibiotics, erythromycin, penicillin, chloramphenicol, and presence of the heavy metals, for example, mercury. This is the next extrachromosomal genetic element after pathogenetic island. We discussed this after the plasmid. The next that is smaller than plasmid is transposon. Transposons in this scheme are shown by the black edicts, black fragments of red plasmid and black fragments of the bacterial chromosome that is shown purple. The migrating elements, and this migration is shown by this errors that demonstrate that transposons, when form their copy, may cause transfer of this copy to the chromosome and integration of this copy to the chromosome, and from one part of the chromosome, these copies of these transposons may be integrated to the other parts of the chromosome. So, transposons are transposable DNA molecules smaller than plasmids. It means plasmids, the large genetic elements, may have circular structure. Transposons and other not, because they're smaller than plasmid and may have only straight stand, non-circular structure. Transposons may serve as the fragment of circular structure of the larger genetic elements of the plasmids and of the bacterial chromosome. Transposons move from one side to another of the same or different DNA molecules, within chromosome, or here, or between chromosome and plasmid, or between plasmid and chromosome, or this last process shown here. Transposons do so by the synthesis a copy of the DNA and inserting the copy at another site. Transposons don't contain information for self-replication. For self-replication, not. Thus, they are not capable of independent replication. The replication depends on the replication of the larger molecule that serves as a container having all these transposons inside the structure. Transposons replicate as a part of the recipient DNA, plasmid DNA or chromosomal DNA. Transposons can code for drug-resistant enzymes, toxins, various metabolic enzymes, occur in several types and copies in the plasmid, cause mutation in the gene into which the transposon is set, alter the expression of nearby genes or their properties. No, that's from the beginning. Here you see circular structure of the plasmid, or maybe chromosome. Transposon. This is an element of this circular structure, linear element of this circular structure. Usually transposons are flanked by the inverted repeat terminals, here and here. In the circular structure, these inverted repeat terminals are shown by lettering. One strand of the transposon has the inverted element with the formula cytosine, 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 adenine, guanine, adenine, cytosine. Between what this strand and strand that is complementary to the first, guanine, 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 thymine, cytosine, thymine, guanine, usually enzyme that is named transposase cause cut. It means what transposase cuts one strand of this double-stranded DNA. After that, bridges between complementary elements of these strands, and after that cut the second strand. It means after function of the transposase, this is and restriction enzyme, transposon becomes flanked by the straight sticky elements that may be complementary and sticky for homologous fragments of the DNA that is cutted by the DNAs in other part, and due to this, transposon may jump from this position to other. It means transposon is flanked by the inverted repeated terminals. Inside has transposase gene, the gene that codes for enzyme transposase, responsible for transfer of this transposon from one genetic element from the chromosome, for example, to the plasmid, or from one part of the chromosome to other part of the chromosome. Exception for transposase genes, transposons have genes for toxins or resistance or enzymes that destroy antibiotics. What this exam demonstrates ampicillin resistance. Also, transposons contain regulatory elements. Repressor, this is a fragment of the gene that codes for repressor protein, and binding site for repressor. It means that this is a promoter, yellow, that is shown here. Practically, transposons that shown here may be transferred because actively produce transposase instrument-to-phone transposition, code for antibiotic resistance, and have regulatory elements that may activate expression of genes in this transposon or may inhibit transmission of genes in this transposon. In this slide, you see better inverted repeat elements, what this, and this, and this, and this, transposase element gene, repressor gene, binding site for repressor protein. We know this binding site, this is a part of the promoter, element of promoter, and ampicillin resistant genes. Ampicillin resistant gene may be replaced by resistance to other antibiotics, may be replaced by gene for synthesis of the toxin, for example, or adhesins or other valence factors. Insertion sequences, IS. They're the smallest transposable genetic elements, smaller than transposons. Insertion sequences have size from 1 to 2 kilobase. In the figure here, you see circular structure of the plasmid. In this plasmid, transposon is present. Transposon is flanked by first and second insertion sequence. And every insertion sequence is itself also flanked by the inverted repeats. First, second, first, second. What do you see here? You see the transposon that is shown here consists of two inserted sequences that are localized in the peripheral parts of this transposon and have the central coding element of the transposon that consists of genes that code for the additional properties. For example, this central element of transposon may code for conjugation, initiation and maintaining the conjugation. If this transposon is responsible for what's coding for this property of bacteria. Insertion sequences have characteristic properties. They have specific sequences at both the ends that are inverted repeats. What inverted repeat? 1, 2. Insertion sequences have a central, what 1 and 2 is, what this is insertion sequence, so this is 1, inverted repeat, this second. 1 second, in the second inverted sequence. Inserted sequence has a central coding gene, what's between these inverted repeats, central coding genes for transposition. It means transposase gene is present here and here. Insertion sequences can cause mutations at their site of integration. Of course, if inversion sequence is not associated, is integrated to the central part of the certain gene, this sequence may cut this gene for first and second elements, and if the gene is transcribed, presence of insertion sequences will start transcription when RNA polymerase will come to the place where IR repeat will appear. And this way, gene becomes non-functional and protein encoded by the gene becomes and now what disappear and becomes functional. But this process of the inactivation of a gene or conversion of the gene to the non-functional state is indeed mutation. And insertion sequences are frequently present in multiple copies at the ends of larger transposons. Well, this situation is shown here. The insertion sequence is frequently present in the multiple copies, with one copy and second copy in the peripheral parts of the transposon, shown here. Variations. So we finish review of the structures, and now about variations, how the structures may be changed. The genotype is hereditary constitution of cell that is transmitted to its progeny and established by the genome. We understand this basic dogma of the microbiology. Bacterial genes may or may not be expressed in a given environment. It means bacterial genes may be transcribed and translated, or transcription translation may be blocked. If translation and transcription translation is realized, protein encoded by the gene is synthesized in the cell. This is an expression. If transcription would be changed, impossible, or transcript would be chemically modified, what expression may be stopped. It means protein in the gene, encoding the gene, may disappear in the organism. Variations of the genome may be genotypic or phenotypic, or genotypic variations are due to the alteration of the genome. They are stable, hereditary, and not influenced by environment. It means genotypic variations alter structure of genome and becomes heritable and stable in the process of the replication of cell. Phenotype, the concept of genotype, is the physical expression of the genotype in given environment. It means that a cell may exhibit different phenotypic appearance in different conditions, and this process is not a result of the changing of the genome. It means phenotypic variations are reversible. Phenotypic variations are not heritable and temporary, and are reversed when environmental conditions are changed. Mutations. Mutations are changes in the coding sequence of the genome. But we understand that mutations are genotypic variations that change coding sequences in the genome. Organisms that carry mutations are called mutants or mutant organisms. Mutations arise by a variety of alterations in the DNA and can be spontaneous or induced. First classification: spontaneous or induced mutations. Spontaneous mutations occur in the absence of any agent known to cause changes in the DNA. It means in the absence of the mutagenic factors that may be controlled. Spontaneous mutations rates are once in 1 million to 1,000 million bacterial divisions. Of course, spontaneous mutations are generated during the process of the bacterial division. Mutation is often the result of errors during DNA replication. Normally, such errors are corrected, but a few escape. Second type of mutations are induced mutations. Induced mutations are produced by agents called mutagens. They increase the mutation rate above the spontaneous rate, not once per million, but once per 1,000 divisions. Known mutagens include chemicals, radiation, and viruses. chemical mutagens are base analogs, analogs of the adenine, thymidine, alkylating agents, and echidine dyes. Radiation, causes mutation. Type radiations are UV ray, ultraviolet ray, X-ray, and gamma rays. Viruses, for example, bacteriophages, when are integrated in the certain part of the genome, that may cause damaging of genes, change the regulation of the activity of genes. They also are, these viruses classified as the mutagens. In the drawing that you see here, processes of the so-called frame shift mutation is shown. With upper line, this is a messenger RNA structure. Below you see gray ovals. They demonstrate chain of the amino acids that are encoded by the triplets of the nucleotides that occur over this what labels with amino acids. Take a look to the central triplet consists of from three uracil molecules. 1, 2, 3. They are purple. This triplet calls for involvement in the protein structure of the phenylalanine. Uracil, this triplet attract phenylalanine and involve this phenylalanine to the structure of the protein polypeptide molecule. Frame shift mutation, for example, may be caused by deletion. It means removing of one of the nucleotides from this sequence of nucleotides. What is happening in this case? First uracil, second uracil are unchanged, but the next becomes guanine. Not the uracil, but guanine. The next base. And this triplet, uracil and guanine, calls for not the phenylalanine, but leucine. The next after the triplet also would be changed and will code for not for lysine but alanine. But absolutely dramatic event would be if you'll see to the triplet that normally code for iline. This codon is converted to uracil adenine guanine. Uracil adenine guanine is the stop codon that stop the process of the growth of the polypeptide chain, and all the downstream amino acids are incorrect or absolutely absent. Of course, protein that what has this mutation, mutated protein, becomes very short and non-functional. And if this is important molecule, for example, cytochrome, this bacteria will die. This would be lethal mutation for this bacteria. Mechanism that you see here is the frame shift mutation. Change of coding frame shift. Changing of all the order of the sequences that code for sequences of the amino acids. Mutations can occur by three types of molecular changes, the basic three types of molecular changes. First, point mutations. Point mutation is the base substitution or nucleotide replacement. They change a single codon in the messenger RNA. Point mutations usually don't cause deletion, it means removing of the nucleotide, or duplication, involvement new nucleotide in the change of the nucleotides. Number of the nucleotides isn't changed, only what some of them become changed. So if point mutation is happened, result may be changing of the amino acid involved with the polypeptide. Normal original DNA, adenine, messenger RNA complementary to the DNA is uracil, uracil, uracil. But 3 uracils code for phenylalanine. Mutation. If third in this triplet adenine is replaced by thymine, the third nucleotide in the mRNA also will be changed to the adenine. And this triplet uracil, uracil, adenine will code for leucine. Leucine would be incorporated in the polypeptide chain, and this would be new incorrect amino acid. This amino acid may cause no death of bacteria by changing their properties, usually. Some mutations don't cause the phenotypic sequences of this type. For example, adenine, if an unchanged third adenine is replaced by guanine, mRNA will have formed uracil adenine and cytosine. Uracil, uracil, and cytosine also code for phenylalanine. It means this mutation would be invisible phenotypically. What is important? Order of the triplets is unchanged. Number of nucleotides in the DNA and mRNA are unchanged. Point mutations. Here you see frame shift mutations. We discussed this. In the DNA molecule, something is happened. In this case, when frame shift mutations happen, number of nucleotides becomes changed. In the certain triplets, for example, here, instead of adenine, adenine, guanine, adenine, adenine, what guanine is removed, this is deletion mutation. And all the triplets, formula of all the downstream triplets becomes changed. As this changing of all the triplets causes changing of all the amino acids in the protein, and finally this protein becomes non-functional. Of course, not only deletion, but insertion also may cause frame shift mutation. To remember, we told that some of the mobile genetic elements are named insertion sequences. If instead of this amino acid would be integrated very large genetic fragment, genetic structure that is named insertion sequence, of course, all the activity of all this DNA fragment that codes for certain protein would be blocked completely. And this insertion, not only of the nucleotide, but of the sequence or transposon in this place, or bacteriophage genome, may cause for these frame shift mutations and switching off of the protein that was affected by this insertion of the nucleotides or migrating genetic elements. And what insertion mutations? Frame shift mutations, we understand what is this. This is a deletion or duplication on the basis of one nucleotide or one triplet. But if larger genetic elements are inserted, these mutations are characterized as a separate type of mutations that is named inserted mutations. And IS sequences are named due to the insertion mutations that may be caused by sequences. But imagine, please, that what is this upper part of the circular DNA insertion sequences is inserted in the central part of the blue gene that occurred here before. This gene codes for the important protein, but insertion sequence integrates to the genome in the central part of the gene and practically disrupts all the function of this gene. Result may be disappearance of the important protein in the bacteria that in principle may cause death of this bacteria. The next group of the genetic variations is named recombinations. Recombinations depend on the process of gene transfer. Due to this, we try to characterize recombination gene transfer in one block. Genetic recombination is the formation of a new genotype by reassortment of genes following the genetic cross. What does it mean, reassortment? It means all the nucleotides or other genetic elements that would be involved in the recombination, they occurred before. This is not the process of the deletion or duplication or frame shift changing. This is reassortment, when genetic structures that occurred before are recombined in the new order and form new mosaic of genetic structures. In bacteria, it involves what recombination. It involves a structural change due to the exchange of the genes within a single cell or exchange of genetic material between two different cells. For this situation of exchange of the genetic material between two cells is shown in the drawing. Upper pink bacterium is not donor that has the molecule DNA, and fragment of this DNA is excised. Below we see yellow bacteria with the blue genome. This is a recipient. If fragment of the chromosome or plasmid of the donor cell anyhow will be injected to the recipient cell, in this time appear conditions for integration of this donor DNA fragment to the recipient DNA fragment. This is not the mutation, but maybe it's mutation, but directly not obviously mutation, because this donor's DNA fragment may cause insertion mutation, may not, if would be situated as an non-coding elements of the genome or between structural genes, function of that is not changed. Transferred chromosomal fragments can replicate unless they are integrated in the recipient DNA. It means to replicate these new fragments of the DNA must be inserted to the larger DNA molecule. Once the DNA is transferred from the donor to the recipient cell, it can integrate into the host cell chromosome by recombination. Integrated, transferred, integrated into the host cell by recombination. Process of recombination is shown in the low part of this drawing when red DNA fragment is integrated to the recipient DNA. Recombination. There are two types of recombinations known, homologous and heterologous. Homologous or legitimate recombination is process in which two pieces of DNA, one of the donor, second of recipient, that have extensive homologous regions pair up and exchange pieces by the process of breaking and reunion. This type, homologous mutation, is governed by the host cell so-called recombination genes that call for enzymes, catalytic enzymes, antiphase, endonucleases, exonucleases, DNA polymerases, DNA ligases of the recipient cell. Heterologous or illegitimate recombination. This process in which little, if any, homology is necessary. It's governed by different genetic laws and requires specialized site-specific recombination enzymes produced by the transposable genetic migrating genetic elements such as transposons, certain bacteriophages. It means not the normal genes of host rec genes are involved, but bacteriophage absolutely foreign or foreign transposons in the conjugative plasmid structure responsible for this heterologous legitimate recombination. Recombination, this is a process that may be realized only after the gene transfer. Transfer genes from donor cell to the recipient, donor to the recipient cells. There are three major elements of the gene transfer known: transformation, transduction, and conjugation. Transformation, this is a process of the breaking of the donor cell, releasing from the cell of the DNA fragment of the donor cell, uptaking of this donor DNA to the recipient cell, and the incorporation or insertion or integration of this donor DNA molecule to the structure of the recipient. It means free DNA that appears in the extracellular space after lysis of the donor cell may be uptaken by the recipient cell, integrated to the genome of the cell. The next, transduction. Transduction requires a special delivery machine. This is bacteriophage. In the transduction, bacteriophage infects donor cell, causes lysis of this cell, and some progeny bacteriophages involved in their structure, in their head fragment of the donor bacterial DNA. If this bacteriophage infects the recipient cell, this phage injects this donor bacterial cell genome, DNA molecule, to the structure of the recipient cell, and together with bacteriophage genes, this part of the genome of donor bacterial cell may be integrated to the large chromosome or plasmid molecule of the recipient cell. And third, conjugation. Conjugation is the process of mating. Conjugative plasmid of the F-positive donor cell contains gene essential for this donor bacterium. This F plasmid codes for peeling proteins synthesis. These peeling proteins form the F pilus that practically serve as a tube, conjugative tube. So that copy of the transmissible plasmid is transferred to the recipient cell and brings to this cell this yellow gene from the donor cell that remains live, not killed, and F factor, red F factor, that makes this recipient cell F positive cell that becomes F positive and requires additional genes from the male cell involved in this mating process. History of discovery of the transformation, the first type of the gene transfer. The site in 1928. Griffith manipulated with the heat-killed smooth pneumococci with the capsule. Heat-killed pneumococci could cause death of the animal mouse. Second bacteria was leaf virulent smooth pneumococci with a capsule. Capsulated pneumococci that could cause death of the mouse. And third variant of this bacteria, leaf non-virulent rough pneumococci that had no capsule, that also couldn't cause the death of the mouse. Griffith mixed heat-killed smooth pneumococci, capsulated but killed, with a leaf non-virulent mutant pneumococci that didn't have capsule. Heat-killed pneumococci by themselves couldn't cause death of the animal. Leaf non-virulent, non-capsulated also couldn't because they didn't have capsule. But if Griffith mixed these two cultures, killed and non-virulent life, and inoculated to mice, mice demonstrated symptoms of disease and die. And after that, when materials from these animals were undergone to the bacteriological investigation, Griffith found in these mice leaf pneumococci, capsulated, was lethal for mice, and leaf non-capsulated pneumococci. And conclusion was leaf non-capsulated pneumococci will not evolve to the recombination to heat-kill bacteria with the formation of the recombinant strains that acquired genes from the heat-killed bacteria and inoculated these genes to the leaf but non-virulent bacteria. And finally, was recombinant strain acquired new capsule and becomes alive. Transformation is the transfer of genetic information in the form of the fragments of DNA or DNA plasmid by means of free DNA or these linear segments of circular DNA by means of free DNA itself from disrupted donors that are not shown to intact and leaf recipients. These cells are recipients. In these recipients, this donor's DNA could be integrated to the genome or could occur in the plasmids, changing properties of these recombinant recipient cells. Pieces of DNA involved in the transformation may carry about from 10 to 30 genes, not very high volume. For successful transformation, the recipient cells must be in a particular physiological state that is named competence. Competence, this is a competence, this is a property of the bacteria in the batch culture that are developed in the late logarithmic stage of their replication. Late logarithmic stage where all the properties encoded in the bacterial genome are expressed, including property to uptake the free DNA from environment and integrated them to the genome of the bacteria. Transduction, the next type of the gene transfer. Transduction is the transfer of portion of chromosomal DNA from one bacterium donor to another bacterium recipient by viruses called bacteriophages. This tadpole virus theoretical structure, the real bacteriophages released from the damaged donor cell. The phenomenon of the transduction was discovered in Salmonella, first in Salmonella in 1952 by Lederberg and Zinder. Transduction can be of two types, generalized transduction and specialized transduction. There are different types of the bacteriophages with the contractile tail, non-contractile tail, without tail. They have all classification, their classification of the morphological characteristics. Now, you have to remember that bacteriophage may establish lytic cycle shown here, or lysogenic cycle that we will discuss later. Lytic cycle, when virus injects the genome to the host cell, after that DNA synthesis of the cell is disrupted, DNA synthesis of bacteriophages is activated. After that, not only DNAs but proteins, bacteriophages accumulate. They serve as sample to the bacteriophage virus particle that after half an hour released from the killed bacteria. So generalized transduction first. This is a type of transduction when any of bacterial or plasmid genes, they're shown as A plus and B plus, this is a bacterial gene of the donor, of the bacterial donor, can be transferred to a new host bacteria by virulent phage. It means it's a generalized transduction, activity of virulent phage is important. You know that virulent phage injects its DNA to the bacterium at here and here, causes accumulation of bacteriophage genomes here, and portions of the uh, donor bacterial cells genes may be involved, included to some of the lytic bacteriophages that would be released from this bacterium to the outside. When a virulent phage infects bacteria and causes lysis of bacteria, that are donors of these A and B genes, any fragments of bacterial chromosomal DNA, any, may be occasionally packed inside phage together with the part of the viral genome. It means bacteriophages in this transaction are virulent, portion of the donor's DNA is any. When this particle infects another bacterium, recipient bacterium, it will bring a new piece of DNA, in this case, piece A plus, new piece of DNA from the former host. And finally, we see the recipient cells that integrate in the genome, fragments that here in the left part of this figure, you see this is shown by the black line with the A plus. In the right part, you see red addict of the DNA to the black chromosome. What has happened? Generalized transduction is caused by virulent phage, transferred maybe any gene or genetic element of the donor cell and integrated to the genome of the recipient cell. Specialized transduction, the contrast. This is a type of transduction where a phage particle transduces specific genes from one bacterium to another. So, phage is lysogenic. Not any, but specific genes may be transferred by these genes. Lysogenic phage infects bacterium. It's usually integrated with specific site of bacterial chromosome. It means when lysogenic bacteriophage is integrated to bacterial genome, after from usually this bacteriophage gene is located in certain part of the bacterial genome with a specific site of the genome. Under several conditions, for example, UV treatment, some chemicals, some antibiotics, prophage, you know that prophage, this is what shown here as a blue, this is a bacteriophage genome integrated to the which can be excised. This excision is shown. Prophage here is shown as a circle blue molecule with the A addict of the donor genome. Under the cell conditions, prophage can be excised from chromosome altogether with the adjacent specific cellular genes. What adjacent specific genes are shown as the A or B genetic elements. When these phage carrying host genes, what carry host gene shown A, what here integrate, involved in the genome of the bacteriophage. When the phage carry host genes, some A plus, infect another bacterium and integrates with the chromosome, the cell has acquired a new progeny, new property that is coded by this A positive addict portion of the genome taken from the donor cell. It means specialized transduction in the contrast to the generalized transduction. Specialized manipulates with the lysogenic prophage or lysogenic phage and specific genetic elements of the donor bacteria that specifically associated, integrated to the bacteriophage genome. No, and the genomes are integrated to the recipient cells together with the red portion of the specific genes uptaken from the donor cells. The genome of that is shown red. Conjugation. Conjugation is the physical contact between two bacterial cells mating during which DNA is transferred from the donor cell to the recipient cell. This is the upper F positive male cell, lower C yellow F minus female cell. The process is controlled by the plasmid F positive fertility factor. Plasmid F positive fertility factor is shown here in the green DNA of the plasmid is a red addict here and red addict here. This F positive fertility factor contains genetic information for the synthesis of X pilus, so this is a X pilus or F pilus, required for conjugation or transfer of the copy of this F positive cell to the F minus cell. Mating begins when the pilus of F positive donor or male bacteria attaches to the recipient female bacteria, F minus. This pilus attaches to the yellow bacteria. Before this process, this recipient cell, F minus cell, does contain F factor. It means can by itself cause a conjugation. The plasmid DNA, shown green, replicates and one copy passes through the bridge conjugation tube or F pilus into the recipient cell. You here see what peel that stated between upper left cell and low left and low right cell. Conjugation mechanism. The process is completed by synthesis of the complementary strand, not complementary strand, to form double-stranded DNA of the effector in both cells. It means process conjugation when it's started. Only F positive cell has the double-stranded F plasmid. After finishing, both the cells have double-stranded F plasmid, and F minus recipient cell is converted to the F positive cell that in the future will actively spread this F plasmid to the other F minus cells in the population of these cells. After that, recipient of a male cell turned to be F positive male, what capable of transferring F positive factor by itself. The F factor itself doesn't contain any different genes. It means F factor itself is free from other genes. So the cell carrying only F positive factor have no features other than the ability to transfer F factor. But in some cases, this F plasmid may be involve other genes, for example, antibiotic resistance genes that code for toxins, and in this case, for the conjugation process, will transfer to the other cells not only F genetic factors possible for conjugation process, but together with these other genetic elements, genes that code for pathogenicity factor or other factors. How this may be done? What high-frequency recombination strains? High-frequency strains arise from F-positive strains when the plasmid is incorporated in the bacterial chromosome. When the high-frequency recombination cells serves as the donor in conjugation, the F plasmid initiates transfer of chromosomal DNA. Usually only part of the F plasmid called initiation segment is transferred, along with some adjacent genes. Because the chromosome can break during transfer, some cells receive fewer genes than the other. The recipient cell won't get a whole F factor and remains F negative after conjugation. It means high frequency recombination. F plasmid integrates to the chromosome and origin site of transfer also as a part of the F plasmid. After that, from this origin part of transfer, so replication of all this chromosome together with the integrated factors happen, and copy from the donor cell start to migrate to the recipient cell, to the F minus cell. Well, the process of the beginning of this transfer is shown here, but in the process of the conjugation, when copy of all the genome of all the chromosome bacteria is involved, usually initiation origin of transfer is transferred partially, and only some cells of the donor cells are transferred to the recipient. After that, usually molecule that is transferred is ruptured, and the recipient cell acquires some new genes with the different functions acquired from the donor cell to the recipient, and only part of the F factor. This part of the factor usually becomes not enough to make this cell F positive, and cell has the non-functional fragment of the F factor and functional genes that were transferred together with this factor to the recipient cell. High frequency recombination gives cells with the increased number of non-F genes acquired from the donor and non-functional fragment of F factor that can maintain conjugation. Other situation is happen when transfer of so-called F prime plasmid is happening. The conversion of F positive cell to high frequency recombination cells is reversible. When F factor reverts from the integration site, it can pick some chromosomal genes. It means F factor, when it's excised from the high frequency recombination cell, this complete functional F factor may pick some chromosomal genes from the donor bacteria, for example, A and B. Such F factor, what this F factor, incorporating chromosomal genes is called F prime factor, F prime factor. The process of transfer of prime factor is named transduction. So, high frequency recombination cell is transferred to the normal cell when plasmid, F plasmid is excised, but during this excision, plasmid may pick some genes from the bacterial chromosome. And later, when process of conjugation is initiated, this F prime factor, this is a factor supplemented with some additional genes, transfers its copy to the recipient F minus cell, and this recipient F minus cell becomes transferred to the F prime cell because it may maintain process of the conjugation and carries double number of the A and B genes acquired from the donor cell and taken to the recipient cell. So, transformation, transduction, conjugation, three types of the gene transfer. Briefly, transformation transfers. This is transfer of the naked DNA from disrupted cell. Donor cell is last. Transformation transfers less than 1% of cell. This is a minimal gene transfer process. Transformation requires competence factor or competence. Only the competent state may be maintained. In the context, transduction transfers is affected by the phage. Specialized transduction, only genes near the prephage are transferred to another bacterium, and lysogenic phage is involved. Only specific genes of donor transfer to the recipient. Generalized transaction is maintained by the virulent phage, and any bacterial genes from the donor cell may be transferred to the recipient cell. And finally, conjugation. Conjugation F1. The transfer is effected by the plasmid F positive plasmid. A single F positive plasmid is transferred, may be transferred without transfer of the additional genes. High frequency recombination. An initiating fragment of a F positive plasmid and a part of the donor chromosomal DNA is transferred. Not complete F1 factor, but part of this factor. Recipient cell remains F negative because F factor is defective, but in the same time acquires new genes coding for other properties different from conjugation from the recipient cell. And F prime transfer. And F positive complete and donor chromosomal DNA is transferred. Process of transfer of this donor genes and of complete F plasmid is named sexduction. No, today's lecture is finished. Thank you for your attendance. Goodbye. Thank you.